Word. Okay. All right. I know we have some people coming in, but we'll go ahead and get started. Welcome everyone to another session of Growing Stronger's A Conversations webinar. We are so excited for this session this evening. We are joined by Daniela Rogoff, who is the Senior Vice President of Clinical Development of at 𝙀𝙊𝙇 Therapeutics. In this conversation this evening, we are going to be discussing 𝙀𝙁's Propel 𝘼𝙊𝙄𝙉 program, discussing the updates, including their 6 months results from their phase 2 data, as well as their plans for phase 3. And again, we are so excited to have Daniela with us, and I know that she has a slideshow for us. So we we'll do that. Before we get started, I did forget. Yeah. Before we get started, as Daniela is presenting and everything, if you have any questions throughout the presentation or throughout this episode, please submit them in either the Q&A or chat box, and we will either answer them as we go through, depending on what works best for Daniela, or if not, we will answer them all at the end. So Daniela, you are welcome to share your screen.
Thank you, and thank you so much for this opportunity, for having us here. This is great. It is good to have these conversations and just be open to answer any questions. So thanks for having us today. It's our pleasure. So let me share my screen. Okay, and now I'm gonna put. Can you see it well, Chandler?
Yes, we're good.
Perfect. Okay, great. So, good evening for everybody that is joining, and thank you so much for joining today. As I mentioned, it's an honor for us to be here and being able to share our work. Having these conversations, we want to make sure that we are able to address questions that you may have. So before I start, I want to say that the safety and efficacy of infigratinib in skeletal dysplasias have not been demonstrated, and infigratinib has not been approved by any regulatory agency for the use described here. So it's important to keep that in mind. It's an investigational medicine, so just that is important to remark. So before we go to the results from our Propel program and going a little bit on what we are doing, I just wanted to take a few minutes to share who we are as 𝙀𝙁 and BridgeBio. So BridgeBio is a company, it's a biotech company that is dedicated to develop novel therapeutic options for rare conditions that are genetically driven, where there is a genetic condition that is or genetic origin that is driving that condition. Our mission is to discover, create, test, and deliver transformative medicine to treat people who live with genetic conditions and cancers that have a clear driver that is genetics. 𝙀𝙁 is an affiliate of BridgeBio that was founded in 2018. It has its headquarters in San Francisco, and currently we are around 50 employees. And 𝙀𝙁 is the affiliate from BridgeBio that is dedicated or focused on the development of infigratinib for FGFR-driven conditions like achondroplasia. So on this slide, what we have is the pipeline for BridgeBio, and this is just to show that BridgeBio is really focusing on rare conditions and developing those hopefully transformative medicines. And all these conditions and these new treatments that are being in development are all in different phases of discovery and testing. And one thing that I wanted to remark is that the model that BridgeBio uses is that for each of the compounds or each of the molecule, it creates a company. So, for example, we are QED and we are developing 𝘖𝙁𝙁𝙁. And this is the intention for that. And the main objective is that it has a group of people that is focused on the advancement of that particular product. So we are full focus and effort is to develop one molecule for one condition or a set of conditions where that molecule could be beneficial. So what is infigratinib? So infigratinib is an orally available FGFR1, 2, and 3 inhibitor that is designed to act inside the cell to decrease that FGFR signaling within the cell. And it's been developed as a mini tablet for potential treatment option in achondroplasia. When I talk about mini tablets, I'm talking about really small size of the tablet that is approximately 2 millimeters. So the size and the being oral administration really is it was keeping or designed or thought keeping kids and families in mind, and to be able to provide an orally and children friendly type of medication. So how would it work? So how do we think that infigratinib works inside the cell? So we know that achondroplasia is the result of a pathogenic variant in the FGFR3, meaning the receptor 3 of the fibroblast growth factor. This FGFR3 acts like as a modulator of growth of the bones in most of the bones in the skeletal by slowing it down. So when that FGFR3 is overactivated and is constantly on an on mode, it's signaling the cells to slow down the bone growth. And this way, the bones grow in a slower way and in an atypical way. So infigratinib is designed to reduce that overactivity inside the cell, inside the growth plate, by acting directly, inhibiting the tyrosine kinase part of that receptor that is constantly or on an on mode. So by doing it so, it is expected that all the signaling inside the cell, meaning all the communication between that receptor to what the cell needs to do at the end, all that signaling is activated, is on all the time. So what we are hoping is that by decreasing that overactivity of the receptor, we could improve the balance between the proliferation and differentiation of those cells that are inside the growth plate, allowing the bones to form in a more typical way and getting not being slowed down like if it would be without the therapeutic, without infigratinib. So after all set of data from non-clinical studies, meaning studies done in animals, where we collected the preliminary or the proof of concept that infigratinib has the potential to potentially provide a benefit in achondroplasia, the program started in the clinics. So currently we have 3 studies that are ongoing. The first study that started, that was started in 2019, is the observational study Propel. This is a natural history study. where all the children that are going to be participating in an interventional study with infigratinib need to enroll first, so we can collect all the baseline data, not only on growth, but on a lot of other endpoints that we want to then evaluate the potential effect of infigratinib on. And this I'm talking about, we are collecting the baseline data on some quality of life questionnaires, on functional abilities, on pain, on the number of medical complications, like surgical interventions. We are collecting a lot of the information in Propel that then we can potentially use in the future studies, including the Propel 2, the open label extension, and the phase 3, to see if we can evaluate if infigratinib provides any benefit or has an effect in any of those very important endpoints that we want to evaluate. So this is our observational study that is enrolling children 2.5 to 17 years old, and that's the first one that we started. So the second study that started is our phase 2 Propel 2 study. This study has as an objective of first provide the preliminary evidence of safety and efficacy. I'm talking about preliminary evidence because the number of children is small and the design of the study was not designed to being what is considered the main phase 3 study. So this is preliminary, all these are preliminary results in a small group of children. So this phase 2 study had that objective to provide that preliminary evidence of efficacy and safety, and also had the objective of identifying the dose that we want to use later on in the phase 3 as a main study to assess the effect of infigratinib during a short period of time, like is in that study that we are now planning. So going back to this phase 2 study, as I said, it had the objective to identify the dose that we are going to use in the phase 3. And for that, the study was designed as an ascending cohort, where each cohort is open after the safety of the prior cohort was reviewed by a group of people, including external experts in the management of children with achondroplasia. Each cohort had a dose that doubled the prior dose. So all the different cohorts that were open was after that group of experts reviewed all the safety data and concluded that it was safe to open the ascending next cohort. So the cohorts that we evaluated were with doses very, very small of 0.016 milligram per kilo. That was our first dose in the study, all the way to 0.25 milligram per kilo per day. That was the cohort 5 dose level. These doses are very, very small compared to doses that have been used in other indications, for example, in oncology. Just to give an idea of the magnitudes of the different doses, the dose in oncology was 125 milligram, and here we are talking about 0.25 milligram per kilo per day. So the study had that after all the children had the opportunity to complete at least 6 months of treatment and safety observations, at that point, there was going to be a data cut just to look at what the results are giving us, so we can evaluate the responses to the different dose levels. And with that, as well as the safety at the different dose levels. And with that, being able to identify the dose that we want to continue exploring. So the other study that we currently have in the clinic is the Propel OLE, that is the Propel Open Label Extension Study. This study has the objective to evaluate the safety and efficacy of the long-term administration of infigratinib. And the children that are enrolled in there will continue to receive treatment until they reach final height or near final height. So this study, same as the observational study Propel, in this study is also a very, very critical study in our program, because this is the study that will allow us to collect that potential response or those potential changes that will take a long time to manifest, and that is not easy to see in a study that has a duration of one year or 18 months. So in this open label extension, we will be able to continue doing those assessments, not only in growth, but on all these other important endpoints that will indicate the potential impact of infigratinib in the overall health. So, so let's go now to the Propel 2, the 6 months result from that dose escalation. So as I mentioned, after all the children that enrolled in all the cohorts had the opportunity to complete that 6 months is when we did this data analysis. So we needed to wait for the 6 months of the cohort 5 to be able to do. So the children that entered in the study and that are included in the analysis are 72 children that are enrolled. From those 72, 39 had completed the study at the moment of this data analysis, and all those children agreed to participate and to continue treatment in the open label extension study. There were 4 children that discontinued prematurely or before the study ended, and 3 of them were because of what it's called withdrawal of consent, where the family decided that they didn't want to continue participating. And these were personal circumstances that would make it very difficult for the family to comply with the study activities. There was 1 child that had to discontinue prematurely because it was going to go. It was going to have a procedure that is not allowed in the protocol. We had, when we looked at the children that enrolled, we had a little bit more females than males. The mean age at study entry was 7.5 years of age, with a range of 3.1 to 11.5. I just want to clarify, the phase 2 study had an inclusion, or the age to be eligible was from 3 to less than 12. Those were the ages that were allowed in the phase 2 protocol. We had a relatively similar number of children that were younger than 8 or older than 8, and we had approximately 17% of the children that were that had a age range of 3 to less than 5. We had also different ethnic backgrounds, and that are described here, and different races. So let's go to, and just one thing that I want to clarify here with the observation of the different or the different races included in the study, was because it's important to have a representation of different ethnic backgrounds and different races, because so that is what we want to have. We want to make sure that the population is representative, not only one ethnic background or one race, but it's a representative of differences and is inclusive of different potential variabilities that we can observe and we can study, and we can give the opportunity to different populations to be included in the study. So having that diversity is very important in clinical trials. So what are the results that we saw? And this is what I would like to share today as well. I'm gonna be describing first the results that we saw on the efficacy, and I'm gonna go I'm gonna discuss the safety in future slides. So the primary endpoint for the phase 2 study is the change from baseline in the annualized height velocity at month 6. That was the main point of the analysis, to be able to evaluate the responses at different doses. What we can see here on this graphic is the change. These bars are showing the change from baseline, and we can see the cohorts 1 to 3 were grouped just because they were all providing, or it was concluded that they were doses inefficacious or provided an inadequate response. The cohort 4 that showed a little bit of an increase of that change from baseline, that in that group that was evaluated, it increased to 1.52 centimeters per year compared to the prior or before starting an intervention. That was change that was statistically significant. But then when we look at the responses that we see in cohort 5, we saw that that change from baseline in the annualized height velocity was bigger than the cohort 4, and that the cohort 1, 2, and 3, and that change was an increase of 3.38 centimeters per year over the baseline height velocity that these children were growing at. So we saw that there was a nice dose response in the change from baseline of annualized height velocity, with a strong response in the cohort 5 dose level. When we see the mean baseline annualized height velocity in the cohort 5 was 3.52 at baseline, and the height velocity at month 6 was 6.9, with a median of almost 7.6. One of the analyses that we wanted to do is to look at a group of children that had a change from baseline of 25% or more. This was because we wanted to try to evaluate those children that had a response that was considered a minimal response that we wanted to see, to think with more confidence that the changes we are seeing are related to treatment, and it's not that we are just measuring variabilities in measurements or variabilities in the growth rate in a period of time. So, and that's why we put this criteria. We wanted to see from all the children who had a change from baseline of 25% or more, and those were the ones that we considered that had a stronger response, and compared to the ones that had a poorer response compared to that one. And this was, as I say, the minimal change that we wanted to see, to try to or to believe with more confidence that the change could be related to treatment. So we saw as well that the percentage of children that met that criteria of change from baseline in annualized height velocity of 25% or more, that percentage of children also increased with increasing doses. And we had that at cohort 5 dose level, 83% of the children, meaning 10 out of the 12 children that participated in the cohort 5, had an increase, met that criteria of the minimal response we wanted to see. When we evaluated those children by themselves, only the children that met that criteria, meaning those 10 children that increased that height velocity over 25%, we saw that those children increased the height velocity compared to the baseline in 4.08 centimeters per year, with a median of 4.1. So I wanted to clarify exactly why we did this analysis, and what it means, that 25% or greater of change. So Other ways, we looked at the data in other ways. One was to see what happens with the 𝙁𝙊𝙄𝙉 score. The 𝙁𝙊𝙄 score indicates where each individual child is located in the growth curve for achondroplasia, for example, like we have here. So as you know, the growth chart has a range of growth of different heights where children can be distributed, and they are distributed from the where the 50% of the children could be all the way from the 3rd to the 97th. It depends on how the growth chart is indicating or is showing. So based on where each of the child is located in that curve, we can calculate the 𝙄 score. It's like how far away from where the 50% of the children are that particular child is. So what we wanted to see is there a change in that 𝙄 score, meaning that position on the growth chart? And we saw that after 6 months of treatment, that position in the growth chart increased on 0.29 SDS, meaning standard deviation score. This is also supporting that those height velocities that we were evaluating also was reflected in changes in the 𝙁𝙊𝙄 score for each individual child. And what we have here is the mean of all the children that participated in those cohorts. Another thing that was very important to evaluate is what happens to the body proportions. We know for sure one thing we don't want is to worsen or make it more disproportionate. But so we were evaluating the changes in the body proportions. What we have here is the ratio between the upper to lower body segment. As it is the upper to lower body segment, any increase of the lower segment that is a little bit more than the increase in the upper segment will translate in that ratio to decrease. So, and this is what we looked and we saw in cohort 5. We saw as well that cohort 1 to 3 didn't result in any change in the upper to lower body segment. Cohort 4, there was a change that was a minimal change. And cohort 5 showed a larger change compared from baseline in the body segment as we measured. These are very preliminary results. These are on a very small cohort of children. This is 6 months, but it's very promising and is something that definitely that is helping us continuing and at least indicating that there is no worsening of the proportion and there may be a trend to improvement. So this was a very promising results. These differences were not statistically significant. Here we have the individual values of the height velocities. We have 2 columns. The dark gray is showing the baseline height velocity, and the green column is showing the annualized height velocity on 𝑖nfigratinib, meaning that height velocity calculated from 6 months of treatment. So what we have is the individual, all the 12 children that participated in the cohort 5. And we see that there is in general a very consistent level of response in the individual children. And as I mentioned before, 10 out of the 12 were considered that had a stronger response compared to 2 where the response, we didn't see that increase in high velocity over 25%. And those 2 children were children, the child number 1 here and child number 4. This, of course, we looked into if there is any characteristics that could indicate why would a child be responded differently, although we know that each child is different and has different tools and sets to guide the growth and response to different treatments. So, but we tried to see if there was anything that was very evident, and we didn't see. But of course, we will need further a larger sample size to be able to do this type of analysis with more detail. But we saw here what we have is that we are showing from the 10 that responded how consistent the response is in general. And as well, you can see that each child is different and it's very individual the way that each child will respond to different treatments. Then what happened to the collagen 10 markers? So all these changes that we were seeing are all clinical, right? All that they can see by measuring the children and by calculating different ratios. But we wanted to see what happens with the collagen 10 marker. That is a marker of bone growth. So collagen 10 is synthesized and deposited in the hypertrophic zones in the growth plate. And once they go through, they get ossified, like they go through the endochondral ossifications. The collagen 10 is degraded and part of it is secreted into the blood. And that is what we are measuring. So as the more endochondral ossification, meaning the changes in the bone to grow, the more it happens, the higher the collagen 10 marker in blood will be. So, and that's what we wanted to measure. We have here the percentage increase from baseline in the collagen 10, and we have the light gray columns are indicating the month 3 results, and the green columns are the month 6. And we see that there is, as we saw before, with increasing doses, there is an increase in that change in collagen 10, meaning that there is more collagen 10 with higher doses. And that is indicating that there is more endochondral ossification happening at the growth plate level. So, and this, the observation of this marker is a good support that the changes that we saw clinically are actually indeed biological changes that that were occurring at the bone level. So now let's talk about safety. So let me go first to this one, and then I'm gonna go back to the cohort 5. So we saw that most of the children presenting at least one adverse event, but most of the adverse events were grade 1 and 2 in severity, and mostly they were assessed as not related to the study drug. I'm gonna show a little bit more of some of the adverse events that were seen in future slides. I just want to remark here that most of them were adverse events that are considered common conditions in children, in pediatric population. We had as well 4 children that had a grade 3 in severity, an adverse event grade 3 in severity. For 2 of those children were children that enrolled in cohort 2, and 2 were enrolled in cohort 3. These adverse events that were grade 3 in severity were ear infections, hydrocephalus, a severe sleep apnea, and worsening of adrenal hypertrophy, as just so we can see exactly the type of adverse events that were seen. Here we have the adverse events reported in more than 10% of the children enrolled. And we can see that these adverse events were considered common conditions in pediatric population, and particularly in children with achondroplasia. As I'm leaving here for a few minutes, so you have the opportunity to look at those. But throughout the entire treatment for all the children, these were the type of adverse events that were reported in more than 10% of the participants. So as we know, we saw that the cohort 5 dose level resulted in a response that was the largest response related to efficacy. So we wanted also to evaluate the safety of the cohort 5 per se, because that was the dose that we are considering, or we were considering for selection, given that it was the one that induced the largest response. And we saw that at the highest dose level, meaning the cohort 5 dose level, there were no adverse events that were considered serious adverse events. No subject had an adverse event assessed as related to the study drug. All the adverse events were grade 1 or 2. There were no grade 3 adverse events. No subject needed or no child needed to decrease the dose due to an adverse event. There were no adverse events at the eye level. There were no adverse events of elevated phosphorus that needed to have a decrease in doses. There were no acceleration in the bone age, also something important to evaluate. And as I mentioned before, there were no worsening in body proportions. So with this, we selected, based on the efficacy and the safety, we selected the dose of 0.25 milligram per kilo per day as the dose to explore in the phase 3 study. So after we completed that dose selection, we continued in or engaged into conversations with FDA and EMA to discuss our plans for the phase 3 and to get an alignment on our proposal study to evaluate the efficacy and safety of infigratinib. We got the agreement that to have a 1-year placebo-controlled trial that it will be randomized in a 2-to-1 randomization rate to receive either active or placebo. So what this means is that we will have a study where 2/3 of the participants will receive infigratinib and 1/3 of the participants will receive placebo. The duration of the study will be 1 year, and the primary endpoint. The primary endpoint will be the change from baseline in the annualized height velocity at 12 months. And we will have secondary endpoints that will include proportionality, the height Z-score, and impact on medical complications and overall quality of life. Based on these regulatory interactions, we are planning to initiate our phase 3 study by the end of this year. So in summary, the safety and efficacy of oral once daily dose of infigratinib at 0.25 milligram per kilo per day will be further explored in the phase 3 randomized controlled trial. The observation lead-in study is currently open for enrollment, meaning that is our study Propel. As I mentioned, all the children that will receive treatment in an interventional study will have to be part of Propel for at least 6 months to collect all that baseline data that we need. And we are planning to initiate the phase 3 by the end of this year. So with this, I just want to give a big thank you to the children, families and advocates that work with us, and that without their help, we wouldn't be able to progress or to have any of the program that we have. So our this research relies entirely on the guidance, dedication, and effort of all of you. So thank you so much, and I'm open for questions.
Awesome. Thank you so much, Daniela. We did get quite a few questions submitted as we went through. I thought it would be best. You were kind of on a roll, so I thought it would be best to just answer them all at the end and through rather than through the presentation. So I don't know if maybe you'll want to go back through some of the slides depending on what the questions are.
Absolutely.
But starting with the first one, and I'm just going to read them as they are. So I heard that there is a phase 3 trial, I guess, in Singapore. Can my child who is currently on Voxzogo participate in the phase 3 trial?
So we are currently including new or engaging new regions, new countries, and Singapore is one of those. We currently, the site, I think it is in the process to get activated for Propel. So that will first, the site needs to get activated and being able to start including children in Propel. The Propel, the plans are always to have the phase 3 as well in each particular site that we are opening Propel. Of course, all of those will depend on the local approvals, right? We need to get, of course, the studies approved by the local health authority and by the institutions. Currently, for our phase 3, and when we will have an update on the clinicaltrial.gov, like hopefully soon to have that description of the study. But for the phase 3, we are still intending to enroll children that have not received prior treatment with other drugs, or either Voxzogo or any other treatment that is in development, as we want to make sure that we can evaluate very cleanly what the potential benefit of infigratinib.
Okay. Thank you. The next one is, what is the timeline for market registration to market application for the drug?
Yeah, so unfortunately, I cannot give an update on that. We are currently focusing on starting the phase 3 and progress as fast as we can. But definitely, we are committed to provide updates to the different advocacy groups, so they can be shared. They can share that information with their family and the community, and as we have it. So we will definitely keep all the groups updated onto that as we get more information.
Very exciting. And I think this one may depend on, various different trials or studies, but at what age can infigratinib be used? So I don't know if they mean quite like when it's FDA approved, or but what's the age groups that it can be used?
Yes, so currently we are studying infigratinib, so it can only be used in the setting, in a clinical trial setting, right? Because it's an investigational drug and has not been approved. All the clinical trials at this point, we are evaluating the children 3 and older. So the minimal age is 3. We have in plans to evaluate younger children, but that is it's gonna be at a different point, at a later stage, but not currently in the clinical trials that we are conducting.
Thank you. How is the mechanism for targeting FGFR3 different from Voxzogo, or I guess vosoritide?
Yes. So let me go here. Hopefully this will help. So we can see here the receptor and the cell, right? The FGFR3 receptor is the one that will have that variant that make that receptor to be constantly on. That variant is that substitution that is highlighted here with the star. That's the change in the protein that makes that receptor to be constantly in a non-mode. Infigratinib targets what is called the tyrosine kinase portion of the receptor. That is the part of the receptor that is involved in translating and all the message from the receptor per se to inside the cell to all the different machinery that needs to occur for growth, ossification to occur or not to occur. Infigratinib then, as it acts at the receptor, it decreases that signaling, that messenger, that messaging mechanism inside the cell. So it decreases all the signaling path that comes that is triggered by the receptor FGFR3. So with this, we believe that we actually can decrease the overactivity of both of the main signaling pathways that are triggered inside the cell after that FGFR3 sends the message. In Voxzogo, there is another. Voxzogo also modulates this pathway, but from a different area. So what it does is, instead of inhibiting the FGFR receptor, and with that suppressing all the signaling pathway that happen below that, it modulates this signaling pathway here, the RAF/MEK/ERK, by modulating the CMP. That is an endogenous hormone that actually is involved in modulating growth. So by doing that, it's also impacting the growth, improving that growth to occur. So it also stimulates the growth by acting on this pathway instead of suppressing the receptor. There are different mechanisms, but they both at the end try to overcome that suppression that the receptor is doing. So infigratinib will work directly at the source and suppressing all the signaling that is triggered from that, and Voxzogo is acting on one of the pathway that is mostly that also is involved in the growth modulation.
Thank you. That was very informative. Are there any updates on if and or when enrollment for the phase 2 open label extension study, I guess, will be, and then, any updates on that? And then they also say, it is my understanding that children who are currently on vosoritide may be eligible for this trial with a 6-month washout, but that enrollment has not been opened yet.
Correct. So the open label extension, the main goal is to continue offering the treatment to children that participated in prior studies, so we can continue monitoring the efficacy and safety until they reach final height. The protocol also accounts for one cohort, that is the naive cohort in the open label extension, that can enroll children that come directly from Propel. And that those children have an criteria for the inclusion or exclusion that is a little bit different compared to the eligibility that we put for a phase 2 or a phase 3, where we need to have a more strict eligibility criteria. So in that naive cohort, we have as a potential of including children that had received other treatments as well, like Voxzogo or other investigational drugs. But yes, that cohort is not open yet. and it is a cohort that will be open upon decision that is needed to get that information.
Okay, the next one pertains to phase 3, particularly from Canada, or the question, I guess, comes from Canada. When will phase 3 begin, particularly in Canada?
Yeah, so we are now in the process of getting the study up and running, and all that will include the submission to the health authorities, to the different institutions. So how fast each of them will be able to open, it will depend on each of the institutions. We know that our target is to start our study in end of this year, but some countries or some institutions may take a little bit longer time to get up and activate it. So it will be it will depend on each country and institution per se.
So I think that kind of like we'll go into maybe the next question a little bit. Is there any information on when the open label study will begin in the UK?
Open label is open and it's enrolling because all the children that are participating in the Propel 2, they after they complete Propel 2, they are offered the opportunity to continue on the open label extension. So that open label extension open as the first child in cohort 1 completed study. So at this point, when we had all the data of the cohort 5 with 6 months data, at the moment the cohort 5 completed the 6 months data, most of the children in the initial cohorts or lower cohorts had completed the study by then. And those already were enrolled in the Propel open label extension as they agreed to participate. So we had 72 children enrolled, and at the moment that we did the data cut, 39 had already gone into the open label extension.
Okay. I think this next question, I know it was submitted during one of your slides. I forgot which slide it was, but it's the study completion is only 39. Did 33 children drop from the study before completion?
Correct. At that time, let me go. So at that time of the data, here. So these 39 were also children that were in the cohorts 1, 2, and part of the 3 by the moment. Cohort 4 and 5 and part of the 3 were still participating. And that's the 72, the 4 that discontinued, and 39 completed. The rest were still actively participated in the study at the moment of the data analysis.
And was there a higher dosage included in the study which could potentially yield better results?
Well, the study, the initial design consisted of the initial 4 cohorts, but the protocol was allowing for additional cohorts to open if there were some criteria that were met. One is that there were no safety concerns, and second, that the a potential of additional benefit from a higher dose, and also that the height velocity at which the children were growing was not exceeding 8 centimeters as a median, 8 centimeters per year. That 8 centimeters per year of the height velocity was kind of like whenever we get close to that, or the median height velocity of a cohort gets close to that 8 centimeter or is above that 8 centimeter, we consider that we reached to what we indicated as our maximum dose that we could keep going up. We wanted to make sure that we were increasing the height velocity, but we were not pushing the height velocity above the 97th percentile of children with average height. That's why we put that median height velocity as our kind of our indicator of how many doses we can keep going up. In the cohort 5, our median height velocity was 7.6, so it was very close to that maximum that we put, and we considered that doses higher than that was going to be exceeding that potential height velocity. Therefore, we considered that per protocol, we had reached the maximum dose, but also we saw that the doses that we were using with the cohort 5 resulting in a very strong response, and it was safe. So between the height velocity that we observed, that was very close to that criteria that we had set as our maximum dose for the study, plus the robust response that we saw in the cohort 5, in addition to the safety, it was what guided us to select the cohort 5 as the dose to move forward.
Okay. And I think this next question comes from the same, I think coincides with that same question, because they both come from the same person, but I think it's like a part 2 of that. So the 2 children that didn't respond, does that mean that the change was less than 25% from baseline?
Correct.
Okay.
So, and here we have their values. So this one, we see that the height velocity did not change in child number 1. And child number 2, it was unchanged as well. However, that child already started from a height velocity that was in a little bit in the higher ranges for the overall population. So those were the 2 children where the height velocity did not increase and did not meet that criteria of the 25%.
Okay. And then I think there's just like 3 more. The first one will be 2 questions because they're from the same person. What is the timeline from the phase 3 trial to market registration and application, and then any indication of the cost of the drug?
Yeah, so unfortunately, I don't have information for either of the parts of the question, but so and as I say, as we get more information and we progress with our program and we get closer to those moments, we will definitely keep our community and the advocacy groups informed. And the prices as well, is that something that I don't have any of that information.
Thank you. And then were there any effects on vision for I guess, you know, children in the trial or enrolled in the trial? Were any of their vision affected, or need for children to wear glasses or otherwise?
No. So we are, as you know, this 𝘼𝘾𝙃𝙊𝙇𝙊𝙀 was used at much higher doses in studies in oncology, and part of the safety monitoring that we are doing, even at these lower doses, are based on some adverse events that we're seeing at much higher doses in oncology. So that's why the close monitoring of many of the aspects, including the eye exams. We have on like eye exams that need to be conducted through our study, our protocols, that helps us identifying if there is any concern with the drug or not, and we have clear rules in the protocols indicating if there is something that is found, there has to be an immediate action with the study drug. So all those are rules that we have in our protocol. We had all the eye exams, and there were no events that could indicate any toxicity at the eye level. There were no adverse events that occurred that could indicate that that 𝘼𝘾𝙃𝙊𝙁 was having a detrimental effect in the eye. And those are not only by the adverse events reported by the investigators, but by all the reports of the eye exams done by the ophthalmologists, where we are collecting all detailed information.
Okay, and then for our last one, since we are right at the hour, and I think it's a good ending question, they start with thank you for your time and work. So I think on behalf of all of the viewers, thank you, QED, for all of your time and work that you guys put into your research. And their question is, would it be possible to extend Propel 3 to South Africa?
Thanks for the question, and definitely something I will discuss internally with the group
Mhm.
as we evaluate the number of sites and regions and countries that we can go. But currently, South Africa is not included among the potential countries, but definitely something I will bring it up to the team.
Thank you. And we understand, as patient advocate groups, we totally understand, how different countries can be, differ from what they can and can't do. But we're always, eager to ask
Absolutely.
that for our,
Absolutely.
for the
Absolutely.
community. But thank you so much, Daniela and QED, just as a whole, for taking your time to join Growing Stronger for a conversation. For those who did not get on or who weren't on at the beginning, this was recorded and this will be available soon. I know when you close out or when I end this, there may be hopefully a little like 8 kind of review questionnaire survey that we would really appreciate if you guys could take like 2 minutes or less to answer. But other than that, thank you, Daniela, for joining us, and we hope to have you back for another conversation at some point.
Thank you, Chandler, and thanks so much for this opportunity. And I will be more than happy to have future conversations with you and the community.
Thank you. Well, with that, we will let everyone go. Awesome.
Thanks.
Bye.
Thanks, everyone.